Okay, so this is really some more of the nuts and bolts of what we've assembled in the Dementia Consortium. We have £3 million available and committed to do this, to generate and advance novel targets. It's been a long time, actually, since there was a new drug for dementia, with the exception, of course, of Lilly, who have stayed the course on this, and other companies are still in. But many companies are actually retreating from neuroscience in general, but this particular area in particular, because it is so tough and because the clinical trials are so incredibly expensive. And because, you know, as we've seen, if you look back over clinical development, there's been an awful lot of expensive failure as well. And so really, we, as Justin, I think, has very eloquently said, we wanted to try and put together a new model for being able to do this effectively, and also to broaden the scope of novel targets and novel thinking, which is always needed in an area. So the Dementia Consortium aims to support pre-competitive collaborative target validation drug discovery. It's a collaboration, as Justin has said. I actually don't really need to go into very much of this because Justin's covered it. But basically, ARUK provides grant funding and oversight. MRCT provides drug discovery resources and the project management. And Eisai and Lilly provide the pharmaceutical insight, tools and potential preclinical drug discovery and development as well, in terms of helping the projects to progress. The projects will typically be up to 2 years in duration, but that's by no means fixed. Different projects, depending on their complexity, will take different lengths of time. And we want to be very, very flexible in making sure that we do the best possible job by any potential project that we receive. Just a little bit about target validation. This is a widely used term. It means lots of things to different people. Ultimately, actually, target validation comes with a successful phase 3 clinical trial, which is often a long way into the future. But in general, I think you can see the signs and symptoms of good targets well before that. So, you know, I'm going to just go through some of these here to stimulate your cortical activity. Preclinically, what this means is, you know, you need to understand, or you need to be compelled to bring forward a target if you can see that it's implicated in disease, and you have evidence for that, or disease models, that you've assimilated a significant database of information to support that. Having a target which is druggable in the sense that there is an opportunity for an intervention on that target to shift its activity in some way is important. I wouldn't be too hung up about that in the sense that so long as it is possible to put together a very good screen, then it is always worth actually just screening to see what you get. So we wouldn't a priori reject a target because no one has successfully drugged it, if you will. It's always worth doing a screen because you can be surprised. Can you find ligands that will modulate the target as desired but avoid off-target effects? Sometimes that's a tough one. But for example, if your target happens to be ubiquitously expressed in the body, then you might want to consider whether you will be able to selectively affect the activity of that target in the brain, if the target should be in the brain, most of them will be, I guess, without causing catastrophic peripheral side effects. We know that actually there are some drugs out there that, or some targets that are ubiquitously expressed, and you can more or less get away with that. But just some thinking about that would be useful, I think. Will the ligands, if you have any, possess drug-like characteristics for further development? Again, don't get too hung up on that. If you have a tool compound that is, if we say, as we used to say in the trade, a bit ugly, so it's got lots of stereoisomers, or it's got lots of phenol rings, or it has anilines and things like that, which generally aren't very good, but nevertheless you've used it as a probe compound and shown good activity, that is a great starting point, because much of that stuff can be removed or re-engineered later on. Can the target be modulated in vivo models of disease? So this will be a critical point in terms of target validation. So in general, when you have established that your molecule can have either a pharmacodynamic effect in an in vivo model, in other words, affect the target, or even better, show some sort of efficacy in some sort of model, which is different from pharmacodynamic effect, that would be great, but that would probably be unexpected. But at that point, if you've reached that point, in general, then there is about a 70% chance that ultimately that drug discovery project will deliver a new molecular entity for the clinic on average. So reaching that critical in vivo validation part is very, very important, and will actually probably be a focus for the Dementia Consortium. And the other thing is, and Mike Hutton has alluded to this, we need to have a sort of a weather eye on what is already in clinical development. So if you come forward, for example, with a novel way of inhibiting BACE, that might not get very much traction because there are already very good BACE inhibitors in clinical development, and the industry has spent many, many, many millions of pounds developing them. So it'd have to be something really remarkable to be able to be competitive with something that's already in phase 3, and therefore between 10 and 15 years ahead of where we